Now I'd like to talk about neurotransmitters in more detail. Neurotransmitters are the chemicals that are released by neurons to communicate with other neurons. And they are important for sort of point-to-point communication. Remember, we have about 100 billion neurons in our nervous system, which make up to, a single neuron can have up to 10,000 distinct connections on it, right, synaptic connections. And neurons are the chemicals that are used to communicate, across each of those, potentially 100 trillion individual synaptic connections. And let's look a little more closely, just as review of all the structures that we've been discussing up until now. So here is our presynaptic membrane. And here is the axon terminal, right? That's where the presynaptic membrane is located. We have voltage-gated sodium channels that are located up here on the axon a little bit before, but when you reach the axon terminal, they are replaced with voltage-gated calcium channels. Remember, we have our sodium current rushing down, depolarizing the neuron as it reaches this axon terminal. The voltage-gated calcium channels open. The neurotransmitter, which is packaged inside these vesicles, right, which is packaged, of course, via what we call this vesicular transporter, this protein that's embedded on the membrane, is going to be released upon the arrival of this action potential into the synaptic cleft, and it's going to diffuse across the membrane, and it's going to interact with various receptors, large proteins, right, that are located on the next cell, on the postsynaptic membrane. Remember, we have two distinct classes, right, of receptor. We have the ionotropic receptors, where the interaction of the neurotransmitter with the receptor is going to result in a change in shape, and it's going to open a channel or pore which will let ions pass through. So this can allow for a change in the membrane potential, right, of the next neuron. It can excite the next neuron. It can bring this neuron closer to the threshold for the voltage-gated sodium channel. It can depolarize it, but it can also inhibit the next neuron, right? It can also, open a channel, say, to chloride, which will come in and hyperpolarize this cell and make an action potential less likely. The other class of receptors, just as a review, are these G-protein coupled, right, or metabotropic receptors. These don't have any pores or holes in their structure, right? But the interaction of the neurotransmitter with the G-protein coupled or metabotropic receptor will again result in a change in shape, right? And this change will result in the release of this G-protein. And this G-protein can then go on to influence other aspects of the postsynaptic cell, right? So neurotransmitters are the means of communication between the presynaptic membrane and the postsynaptic membrane, and the interaction of these neurotransmitters with their receptors is key to their influence on the next cell.